Thank you all for being here. My name is Erin. I am the Life Sciences Lead on the AWS Open Data team, and I am very excited to be introducing some fantastic talks from people who are managing truly game-changing data sets in the life sciences. We did put the word life sciences into this title because these are very life sciences focused data sets. We will be talking about genomic data and connectomic data. However, I would encourage you to keep kind of an open mind. For those of you who are here for the open data and not the life sciences, remember that under the hood, data are just bits and bytes. And so when we talk about unstructured data or volumetric data or text data, unstructured data, these are all principles that can be applied to any industry. But let's zoom back a little bit and talk about, the value proposition of open data. The fact is we know that data are getting bigger, and as data gets bigger, it's very hard to keep those data siloed and be able to draw relevant insights across many data sets. And so what we've proposed is the research community can actually benefit from large single data repositories that help to harmonize and aggregate data coming from lots and lots of researchers so that the community can utilize those data in mass. And we know that this is beneficial not just due to the scalability of things like cloud-based storage. We know that if you can lower the cost of being able to store data in several places, you ultimately lower the cost of research. You can also encourage use of open or conventional formats so that people can use data the way they want to use it, apply as many analytic engines as they they choose, and ultimately potentially go beyond insights by applying things like AI or machine learning. So our mission on the Open Data team is to make more data available to more people. Ultimately, if you centralize high-quality, high-impact data in the cloud, we believe that you can reduce your time to insight, lower your cost of research, reach a global community of users, and since your data are already in cloud, you have those brand new tools and features right at your fingertips. But I want to ask you an important question. What happens when your data and your user community are big? You can't move it, you're right. And at the same time, you have lots of people who want to access those data at the same time. This is something that we're running into in the life sciences over and over again. Our data are approaching petabyte scale, and our user base ranges from the thousands to the millions of researchers all over the world. So today, I'm very happy to introduce two, let's call them sentinels of data sets that reach that petabyte scale and are reaching thousands, millions of users to draw insights and make their science happen. So without further ado, I'd like to introduce Dr. Rodney Brister from the National Library of Medicine. We do have a little bit of time to take questions after each of our speakers. So after Rodney speaks, he can take a couple questions from the audience, and then I'll introduce Dr. Brock Wester from the Johns Hopkins University Applied Physics Lab. So without further ado.
Thank you for the opportunity to talk today. During this presentation, I'm going to describe some of our early steps at the National Institutes of Health to build a public biomedical dataverse in the cloud using data that the institute I work for, National Library of Medicine, oversees. The National Library of Medicine is building a vast public biomedical sequence data set called the Sequence Read Archive. The SRA is a collection of user-submitted DNA and RNA sequences and is the world's largest publicly available genetic sequence data set. The archive supports a number of scientific use cases, including investigation of genetic variation and gene expression, as well as exploration of microbial communities and the identification of unknown species, viruses, and genes. The SRA is a community repository, and data submitted by individual data generators and research organizations are aggregated into the archive. More than 22,000 individual entities submitted data to the SRA in 2021, And these submissions include both genetic sequence data as well as descriptive metadata that includes information about how the sample was obtained, any clinical or environmental conditions that co-occurred with the sample, where it was isolated, and information like that that really makes the search and analysis of the data doable. The SRA stores both open access data, which is going to be the focus of my talk today, as well as some controlled access human data that's available to authorized users. So the SRA supports fair data practices. That is, the data is findable, accessible, interoperable, and reusable. To this end, sequence data is submitted to the SRA in a variety of different formats by the data generators, but it is transformed into a single SRA normalized format. This approach ensures the data in the archive is interoperable across all the different submissions over time and facilitates the reanalysis of previously submitted data. This data is also searchable and retrievable through several NLM maintained resources and tools. And metadata packages are available through the biosample database to support community standards. So if communities, individual data communities, have standards about what metadata should be included with their samples to make those samples more accessible and more usable in their workflows, then we can support that. The SRA's data set is quite diverse, and it is really derived from across all the kingdoms of life. This includes genetic sequences from humans, research model organisms like rats and mice, agriculturally important organisms like cows and pigs and wheat and corn, as well as surveys that are being used to determine the biological diversity across the planet. The archive also includes so-called metagenomic sets, and these are derived from environmental surveys as well as microbial surveys. You may have heard of these as microbiome studies, the microbiome of your gut or your skin, or the microbiome of root systems. These have become increasingly important to understanding the biological systems on our planet, as well as interactions between like human health and the microbes that live in our body. As you might imagine, data submission and usage changes over time as certain scientific topics get hot and the various scientific use cases change and adapt to that. One salient feature of the SRA is that the value of the archive grows as more samples are submitted to the repository. There are multiple data creator communities, as I described, each creating sequence samples in response to their own use cases, and multiple data user communities, each using the data in response to their own use cases. And as you might imagine, data submitted by one community being used by another community means that people are using the data in ways that the initial community never imagined. And this is a pretty amazing thing about the SRA, and it really underscores a key point. The data is reused. So data submitted 10 years ago, 5 years ago, continues to be reused today. Ever-changing use cases, always wanting the big aggregate data, and people go back and pull those old data sets out and compare them to what's being obtained today. And then you can imagine the chronology of data is really important for a lot of studies. Studies about the environmental fitness of a lake or a river, or the evolving nature of viruses infecting bats or other known zoonotic species. So the SRA has grown really rapidly over the past decade, and a single copy of SRA now includes 19 million unique data samples, or over 20 petabytes of data in the SRA normalized format. The SRA data set is not only very large, but it's actually used quite extensively. And indeed, more than 51 petabytes of data were downloaded or egressed in 2021. So 2.5 volumes of the archive were downloaded or egressed last year. An amazing sum of data usage. Although the growing size of the SRA increases its potential scientific impact, it also hinders the sustainability as well as the usability of the archive. So the bigger it gets, the bigger the potential impact, but also the bigger the problems encountered in trying to use it. This problem is actually not unique to the SRA, as Aaron alluded to, and NIH has launched an initiative called STRIDES that seeks to leverage commercial cloud providers in order to create an equitable and sustainable ecosystem where NIH data is fair. NLM began replicating the SRA as part of the STRIDES initiative in 2019. And also probably of note to this meeting today, as we began replicating the SRA and interacting with the various cloud service providers, we also began interacting with Aaron and the AWS Open Data Program. Initially, we were focused on the COVID data sets that were in large demand at the time, but by the spring of 2021, we'd actually replicated the entirety of the public SRA onto AWS ODP. This program allows not only for free storage of the data for NLM and NIH, and quite frankly, the American taxpayers, but also provides free egress and download of the data for both cloud and non-cloud users, thereby providing equitable access to the SRA by a variety of users. And this is really critical to us because we want to have an equitable resource, not just sustainable, but also equitable. Replicating on the SRA on the AWS ODP has had a profound impact on our usage, as you might have imagined, and we now see an average of 8 million data samples used each month. Again, an enormous sum of data. So what are people doing with all this SRA data? One example is the Ceratus project, which used AWS ODP hosted SRA data to identify novel viruses and previously submitted samples. So again, we're going back to data reuse. The Ceratus project examined 5.7 million individual SRA samples, more than 10 petabytes, a feat that would have been very difficult had they been trying to download this from an FTP site, right? They were able to leverage the cloud analysis tools that they were using, and as many as 1 million samples per day were being processed. Again, a huge sum of data. So, end result of this, they identified 100,000 new viruses that had not been identified before. Now, they've gone on to build a web interface that connects lots of users of different varieties to this back end, and allows now people to use their cloud-hosted tools and data to do amazing things without having to know how to engineer in the cloud. So what are we trying to do by moving the SRA to the cloud? Well, first of all, we're trying to build a sustainable ecosystem of data and tools that is accessible to bioinformaticians, data scientists, solution engineers, public health scientists, graduate students, and librarians, a huge swath of users from a diverse range of institutions, both well-funded institutions as well as those that are not so well-funded, that supports petabyte scale use of the data. We want the archive to be leveraged in its entirety and encourages federation of data and metadata from other biomedical data sets. That is, the SRA doesn't exist in a workflow all by itself. In a typical workflow, people want to connect SRA data with other data, and we need to facilitate that in the cloud, and ultimately to serve a broad range of biomedical use cases. So our approach to building this ecosystem includes several areas of focus. First, we realize that many of our users are not currently working on the cloud. So we are developing educational materials to help onboard users. We're also engaging them directly with the scientific community through codathons, workshops, and webinars. We use these and other engagement activities to document use cases and really understand the scientific goals of the folks involved. And this is really critical. We want to build something built for purpose. If we can build something that's actually useful for the scientists, that will give them some incentive to go and start working on the cloud, figuring out how to move their workflows to the cloud, and actually use it. If we don't build something built for purpose, who cares? Finally, we're identifying tools that are critical to the successful ecosystem and in prototyping solutions. So one prototype solution is our cloud-hosted SARS-CoV-2 sequence analysis pipeline that was built by NCBI as part of the Active Trace project. This pipeline calculates viral mutations across all samples of SARS-CoV-2 that have been submitted to NCBI. And we've been working with industry and public health partners to develop best practice and refine our analysis. To date, mutations have been identified in 6.3 million unique SRA and GenBank samples over the course of about 15 months, i.e. the course of the ongoing pandemic. And this data is available through both the AWS Open Data Program as well as Google Public Data Sets. Our SARS-CoV-2 analysis is really creating a new paradigm in pathogen data. Not only are we making data available through analysis files that support high volume of visualization of the underlying data, but we're also providing our analysis results through Athena and BigQuery. This arrangement's really kind of cool because it allows a data search and analysis based on the mutations, quality metrics, and other descriptive metadata that we either calculate or are included with the sample, and essentially supports a very large-scale computation pathway using simply SQL queries. So you can define all the parameters of what you want to investigate through the SQL queries and then interrogate these big tables with all this data to find the answer. As I mentioned, all this data is available through the cloud, and we certainly encourage the use by everyone. Some of our current users include Galaxy and Chicagoland COVID-19 Commons. And of course, we love platforms. We love third-party platforms. We would love for people to be accessing this kind of data and making it useful and available to a broader range of users, not just folks that know how to do SQL queries and BigQuery and Athena. NCBI is also developing several cloud-native tools to analyze cloud-hosted data. This includes the Read Assembly and Annotation Pipeline tool, or RAPT. This pipeline assembles raw bacterial and archaeal sequences and then annotates their genes and proteins. And this is really sort of your typical workflow that a bioinformatician might do. They sequence a sample, and then they take that raw sequencing data, they assemble it into a genomic sequence, and then they go and identify the genes and proteins there. And so this is kind of our core thinking here, just to provide these basic elements of a workflow that people can get their feet wet using the cloud. Another thing we've done here is actually created a web interface that allows people to interact with this. So you don't actually have to use this in the cloud. You can go through our web interface and use the same tool with the same speed and the same benefits that you would find using it on the cloud. We've also worked on developing a tool called Elastic Blast, which is a cloud-native version of one of the most popular sequence comparison tools ever created. And again, trying to move these essential tools to the cloud and start onboarding users through them. Finding SRA samples of interest is critical to using SRA data, obviously. And there are really two basic types of searches. The attribute searches, where samples are queried based on the text and their descriptive metadata. And then sequence-based searches, where samples are queried based on their nucleotide sequence itself. This latter approach is really central to many bioinformatic workflows, and efficient methods to search for specific sequences across the entire petabyte scale SRA are really needed to make this huge archive accessible to a lot of users. To tackle this, what is quite a difficult problem, we have formed a cross-agency partnership with the Department of Energy. And through this partnership, we've been identifying use cases and trying to drive some solutions. As you guys may remember from one of my earlier slides, metagenomic samples comprise a large percentage of SRA corpus. And these samples capture the diverse microbiota present in our bodies and other ecosystems, and are really becoming increasingly critical to understanding aspects of human health and the emergence of pathogens. The organismal content in these samples can be complex, and quite frankly, may not be known at the time of sampling. So you may sample something, go looking for one organism, but there may be 10 other organisms in there. And as I alluded to before, some of these may not even been identified. So it's sort of what we like to call the known unknowns. We know there's some stuff in there, but we don't know exactly what it looks like. And so working with DOE, we've developed a series of workshops and codeathons designed to engage the scientific community, document their use cases, and produce prototype software that allows people to take a sequence of interest, something they know about, and go find things that match that across the SRA corpus. An upcoming codeathon is actually scheduled for September 2022 to produce working cloud-hosted solutions. And as the announcements would go out, we invite participation by the community. We're interested in both scientists as well as solution engineers working in the cloud. As I mentioned before, many of our users are not yet working on the cloud, so it's critical that we continue to work towards onboarding the scientific community. This is a place where we can work with other stakeholders and members of the scientific community. I mean, we can't feasibly make all the how-to documents that people need. And we're at this point reaching out to the rest of the community going, let's work together to really help people find solutions in the cloud. We also can't build all the tools and ourselves. There's some tools we can definitely support to keep things moving, but we need to work with other stakeholders to ensure that critical tools are available that enable compute across the petabyte scale SRA. Finally, we need to connect SRA data to other data sets, and it's not just raw data, but analysis data and third-party data platforms, so that users have access through the descriptors on a particular sample or sample IDs to link together different data and complete their workflows. So, as you might imagine, this is a huge endeavor and required lots of people. I'd like to give a special thanks to all the folks at NCBI, particularly Yuri Skopchenko and Denise Hicks as part of the SRA team, and then Kurt McDaniel and Ravinder Eskandari as part of the STRIDES leadership team. Lots of other folks around NIH and at DOE that have helped all of this come to fruition, and quite a few really important partnerships that have really been important to making this all happen. Thank you.
Questions for Rodney? Oh no, we have microphone runners.
Yeah, the National Cancer Institute had the Genomic Data Commons at the University of Chicago. Is that still there, or is that a data set, since it's petabyte scale, genomic data, something that could be moved in with you guys?
So that's a really great question, and it gets into two aspects that we're now working on. One is the controlled access human data, which we've also moved to AWS ODP. And then two is the way the system in controlled access human data across NIH-funded activities is actually already federated. So that is to say that the raw data exists in multiple places, and we're working through NIH through a program called NCPI to try to link that data together. And so there's access and authorization services that are being developed to help do that. We're also talking about using things like a database hosted at NCBI called dbGaP, which includes a huge amount of metadata that's been transformed, using things like dbGaP to create what I like to call the index of everything, where you can go to one index and do your sort of text-based searches, and then you'll find the URL connected to the data, no matter where it is.
I think we have time for one more question.
Thanks for the great presentation. You mentioned you're participating in the ODP program. Are you doing that just for the publicly available data sets? You also mentioned controlled access data.
Yeah, we're moving the controlled access data now, and it's, I believe, already there.
I think so.
So all of the controlled access data is also part of the ODP program?
Correct.
Okay, great, thank you.
I think we could do one more. That was a quick one.
So you mentioned a big part of this was educating people on how to leverage like cloud compute in order to leverage this data set. So like what are some techniques you see people use? Are they like spinning up individual EC2 instances or deploying like EKS clusters? What do you notice?
Yeah, so lots of questions there. So some things, there's a sort of working in place idea there in the cloud right now, which means the data's located one place and my compute's located someplace else. So what folks are doing is not quite real-time streaming. They're moving the data to where they're working, evaluating that data, analyzing that data as quickly as possible, and then throwing it away. So I think there's this concept that this kind of approach, when done very efficiently, can help support folks no matter where they're working, no matter what their cloud environment, as long as they can pull that data quickly and efficiently, can support them no matter where the data is. And I think this kind of gets to this big point that we're trying to solve right now to really satisfy the needs of efficient compute. How does the data have to be staged? Can you put all the data in one place and then support efficient compute by this computation in place? Or do you actually have to store the data in various places? And I think there's still some questions around that. And it gets back to equity and access and all these sort of other topics that we're trying to really sort of hold as goals for our activities.
Yeah, that was a great question. And I also will add, we are going to move on to Dr. Brock Wester, so he gets his 20 minutes. But I did find out that this session is the last one before lunch. So if you want to pepper them with questions, I'm throwing them to your mercy after the talks are over. So without further ado, Dr. Brock Wester.
All right. Great. We will look forward to that, I think, the peppering part. So I'm really happy to be here. My name is Brock Wester. I'm a manager and a researcher at the Johns Hopkins University Applied Physics Laboratory. And I'm going to talk to you today about the future of petascale neuroscience and how big data neuroscience enabled by things like Open Data Program are going to make impacts in medicine and science. So I'm going to start by talking about the BRAIN Initiative, which is a 10-year, multi-billion, multi-sponsor, nationwide initiative focused on revolutionizing neuroscience research. And a big part of this initiative is investing in the development of tools and technologies to enable the next generation of neuroscience experiments and studies to enable more neuroscience discoveries. And a lot of this is centered on data. The amount of experimental data that's being generated is increasing exponentially. And there are new tools for imaging larger extents of the human brain, of animal brains, with higher resolution. There are new tools for recording neural activity in more and more locations all at the same time. And so with these new tools and technologies in place, the field can perform new enabling research studies that can improve our understanding of the brain and give us insights into the cells and circuits that give rise to movement and sensation and development, and that are a part of the mental experience, our behavior, and our well-being. So at the center of much of this work is data, clearly, collecting it, storing it, processing it, making it accessible, all to enable science. And the NIH and the BRAIN Initiative right now are in facilitating what I think is a paradigm shift in the neuroscience community, in that they're requiring all funded research labs to actually make their data publicly available to the rest of the neuroscience community. And a key part of that is this brain informatics program, which is focused on developing new data archive technologies, new processing tools, and new data standards, all with the thought of making neuroscience data fair, findable, accessible, interoperable, and reusable. And each data science community that you see here on the right has their own challenges. They all have their own metadata schema and different workflows. And in some cases, the different data communities are focused on basic research or could be focused on research that makes an impact in medicine. And so we just heard about the impact that genomics is making in things like the pandemic, where genomics research is enabling the development of new vaccines and new ways to monitor virus evolution and testing and clinical care. But today I'm going to talk about connectomics and high-resolution neuroimaging data sets. So connectomics is a study of connections and connectivity in the brain. And this can be high-level connectivity. Think different regions of the brain, like the visual cortex, which is responsible for processing visual information, and how that connects to the part of the brain that plans for movement. But connectomics also goes all the way down to the lowest level, the single synapse, which is the physical connection between two brain cells or two neurons. And their connectomics, like genomics, is all about creating maps which can give us insight into connectivity, how the brain is wired, how it collects and processes information, how it stores memories, and how it plans actions. And in the human brain, there are 100 billion neurons, thereabout, and each neuron can be connected through thousands of different synapses or connections. And developing the means to map this circuitry is really critical to enabling science. So lots of potential for impact with this space. It's considered to be, by some, a missing link to developing diagnoses, prognoses, treatment, and even the reversal of numerous neuropathologies and diseases. And also understanding the brain, which is very good at processing information efficiently, could be useful for developing new means of artificial intelligence and machine learning. And given the power efficiency, which the brain operates at about the wattage of a light bulb, there's a good opportunity for low power, low weight, low size electronics and robotics that can be derived from neural architectures. So just to give you insight on what this looks like, the single synapse connectivity maps are generated from neuroimaging volumes. And it all starts with the synapse, which is so small, tens of nanometers, that it's smaller than the wavelength of light. It can't be visualized except with electron microscopy. And so this video by Bobby Kasturi and Daniel Berger and Jeff Lichtman at Harvard does a good job of highlighting what these data look like. And it starts with the image volume, which is segmented to identify the cells, which are the blue and the orange paint there, and the synapses, the connections between the cells. And from these, we can derive maps. Think of these maps as like balls and sticks, basically can be represented by a mathematical object called a graph. So the microscope systems that are in place to generate these kinds of data sets are able to collect data orders of magnitude faster today than they were just a few years ago. And so that presents a great opportunity for science. But as everyone here is thinking, right now, there's also some challenges there with the size of these data sets. And so these data, in some cases, are so large that no human being will look at them. But they can be processed by computers to identify the cells, to identify the synapses, and other features, neuroanatomical features in the data sets that can give important scientific insights. And while this growth is exciting, it was clear some years ago that the existing file systems and data systems that were in place were not tenable for dealing with this scale. And so we need something that would scale on demand for ingest and processing and the future of this field. And so as a part of the IARPA Microns program, our team developed the Brain Observatory Storage Service, which is a 4D, 3D neuroimaging data ecosystem. And the IARPA Microns program was focused on reconstructing or reverse engineering a cubic millimeter of the visual cortex of a mouse. And this was one of the first contiguous petascale image volumes that was generated, orders of magnitude larger than any of the volumes that had been collected by the community at the start of the program, which was in 2015. So BOSDB can store those kinds of image volumes. Electron microscopy, X-ray microtomography, very well suited for high resolution neuroimaging data sets. So it'll store the image volumes themselves. It'll store the segmentations and annotations on top of those image volumes. Think the blue and the orange paint. It can store the meshes and other data derivative products that are generated from the original imaging data. But it also stores the connectomes, the graphs, the edge lists, the connections between the different cells, which is where some of the power for future science is. So there's a lot under the hood. Our group's given a few talks and AWS events to highlight some of the technologies, and I won't go into too much of the details here, but our system is heavily enabled by AWS services, from highly available data storage and data access to scalable ingest. Think something on the order of like hundreds of terabytes over a few days. Data processing over contiguous petascale image volumes for downsample and segmentation and things like that. And so it's obvious that AWS is really enabling here, and we also rely heavily on serverless capabilities. So because these image volumes are so large, and in many cases there's just a few processes that need to be run at one time, we can scale up massively, run that process, and then scale down as needed. So it's a lot of hurry up and wait kind of operations, and serverless is really great for ensuring performance and doing it efficiently and at low cost. So the BOSDB system is storing 36 public projects as a part of the AWS Open Data Program. And these include the image volumes and the segmentations and other data products associated with these image volumes. And we also collect a bunch of metadata that can be searched and be made available to enable different scientific workflows. These data sets are made available through an international CC BY 4.0 license. And we have a bunch of data sets from the BRAIN Initiative, but also data sets from the community going all the way back to 2011, which were originally collected by Neurodata, which is a collaborator at Hopkins. So the main goal for BOSDB, as with other data archives, is for enabling science, which aligns very nicely with the AWS Open Data Program. And we store data sets from a variety of organisms, just like Rodney mentioned, different invertebrate systems and mammals. And it includes the image volumes, the segmentations, and all the various data products, including the connectomes. And we have different parts of the vertebrate and invertebrate nervous systems represented by all these different volumes, different sizes, different extents, and in some cases, different imaging modalities. But each of those can be accessed through the same API, and we can get data in a variety of different formats, subvolumes or image tiles, objects, and so on, through the scalable API. So I thought it'd be important to highlight just the impact the great impact that the AWS Open Data Program has had on BOSDB. So this just shows our open data bucket over the past few months. And we actually just completed an ingest of another data set from that same IARPA Microns program. This is a 100 terabyte contiguous image volume of mouse visual cortex. It's also worth pointing out the numbers of objects, data objects associated with these data sets. That's the pink dotted line, in some cases tens to hundreds of millions. And so serverless technologies are critically enabling for moving these things around, processing them, and making them available in a scalable way. So I mentioned our metadata service earlier, which is something that's available through a command line interface for querying and searching. We also are exploring some natural language querying capabilities, and this is something that can provide users with good insight into the kinds of things that could be learned from these data sets. Simple questions, things that you guys are probably very curious about, like how many excitatory neurons are in this volume? Or how many mitochondria are there next to this synapse number 904? So we can provide those kinds of data outputs. And queries run against these kinds of massive data sets can allow the scientific community to get a better understanding of how the brain is connected, how it's wired, how it's organized, and all those insights can be valuable for medicine and science. And in the future, we're not quite there yet because these image volumes take a lot to collect. But in the future, we're going to have data sets not just from healthy animals, but also from diseased animals, animals that have neural injury or have developmental disorders. And by comparing the connectivities between those different data sets, we can get insights into how disease manifests and potentially how therapies can be applied at different time intervals along the progression of a disease. So connectivity, understanding the connectivity is a big part of this. So we've developed a lot of tools that can mine and search these connectomes, these brain maps at scale. And these tools are really necessary because the numbers of connections that we're talking about are pretty significant. The IARPA Microns program, the largest data set, has hundreds of millions of connections. And so there's a lot of data packed into those connections. Being able to run through that and identify patterns of connectivity, things we call motifs, which we think are important to how neural systems work, is a very computationally expensive process, but we've developed tools that enable that and make it feasible and scalable. All right, so our team is really excited about how far the field of connectomics has progressed in recent years. And with the increasing amount of resources being provided by the BRAIN Initiative and by AWS, we're really excited about where this field is going. So you just saw the impact of the genomics field from Rodney's talk, and the genomics field makes an impact on humans every day, especially from the pandemic, when you think about all the things that have come from that. And in many ways, the capabilities today were enabled by the Human Genome Project, which was an initiative started 30 years ago. And it's made a lot of impact in the technologies that are present today, that are available to many researchers today. And it started with mapping the genomes of very small organisms like worms and flies, and then moved to rodents, and then to humans eventually. And there are many in our field that think that what's happening in the connectomics field is following a similar progression. And so we're really excited about the future. And in fact, the NIH right now is getting ready to launch a new program called the Brain Connects program, which is going to provide resources for the next series of brain mapping data collections, and in a way following this playbook set in place by the Human Genome Project. So the next major goal of this Connects program is to develop the enabling capabilities that will allow the mapping of a whole mouse brain, which is 1,000 times larger than the fruit fly, which was mapped just a few years ago. And of course, data solutions are going to be key to enabling those experiments, those data collections, and the future for this field. So many say the goal of mapping the genome in a way has been realized. Not that the work is done, there's still much to do, but the impact is already enormous, developing cancer treatments and new therapies and treatments, and obviously vaccines for the pandemic. And the Human Genome Project was a big part of enabling all of that. And it was a key motivator and inspiration for the BRAIN Initiative. That's providing targeted investments in how we will perform the next series of neuroscience experiments. Investments in infrastructure and data ecosystems, all with the thought of revolutionizing this field and enabling the future therapies that will impact us down the line. So it's very, very exciting. I want to thank you for your attention. Also want to thank the BOSS-DB teams and the IARPA Microns program teams. This is the Princeton University Allen Institute for Brain Science and Baylor College of Medicine, as well as some of our collaborators. And of course, the NIH and IARPA, who made a bunch of funding available through the BRAIN Initiative to enable what we're doing and to enable the field to grow and make impact for the future of science and medicine. So thank you.
Questions for Brock?
Hey guys, do you guys version control your data?
Do we version?
Version control your data? Yeah.
Is that right? You're asking if we version the data sets?
Yeah.
Oh, okay. So in many cases, we will publicly release data sets that have already been processed using a particular workflow, but the data sets are there to be reprocessed, and so users can create additional channels, and that can be overlaid on the existing original raw image data, and so they can create new segmentations and new annotations and things like that. And there's no limit to the number of channels. We've got thousands of them. But in terms of versioning, in many cases, that can be enabled by a graph database system, which I can talk about a little bit later. And that's more focused on the connectivity and the segmentation. So there are systems that we do use right now as a part of the IARPA Microns program that are part of the proofreading effort, which is scouring through this data set and making changes to the segmentation to improve mistakes that are generated from the computer. And so this is a system called PyChunked Graph, originally developed by Princeton University. And so we can leverage that system in collaboration with them and other collaborators to make updates to the Microns data set. It can be leveraged for other data sets. But yes, there's a way to do that. PyChunked Graph allows for a lot of querying at different time points and at different version numbers, and can serve up data in a variety of different ways. It's a great resource that we use. We plug into a lot of different community tools for versioning and for other things. Thanks.
I think we have time for one more question. Okay, I have a question then, Brock. So I'm a genomicist by trade. I should introduce myself. So I'm an animal genomicist, actually. And so when we think about identifying a missense variant in a gene, we can think, well, the immediate therapy would be potentially to replace that gene or replace the protein that gene codes for. And so are there examples in connectomics where you're directly targeting the connectivity of the brain and be able to show that impact in the clinic?
That's a great question. So the field right now is so early where those kinds of experiments aren't quite possible at the single synapse level. In many cases, it can be used as a means to get insight into what might happen during the course of a disease, and then, of course, what could happen as you apply a therapy at different time points. And unfortunately, in many cases, the data sets I highlighted here, you've got to slice up the tissue to get insight into the connectivity. There are other means of connectomics for functional magnetic resonance imaging. But in terms of getting those time points, it's really enabling as a research tool. And in clinical practice, it won't necessarily be available until we get the next imaging tool that will allow for that kind of image resolution in live humans. But yeah, this can be done in animal subjects with various kinds of experiments.
Thank you. One more hand for Brock. Okay, so I'm just going to do a quick plug. You saw a couple of screenshots of the Registry of Open Data on AWS on both Rodney and Brock's talk. This is a digital catalog of available data that we know exists on Amazon S3. Actually, last time we checked, we helped to distribute over 100 petabytes of open access data via the registry, so please go ahead and check it out. We also have a number of usage examples, tutorials, and publications that demonstrate how people are using the data. So I invite you not only to look at how people are using the data, but get some ideas of how you can use it yourselves. And the other thing is I want to hear from you. We are a relationship building team as much as we are a data team. And I want to hear what you want to talk about, how you want to use data, what data sources are important to you. Please reach out to us at opendata@amazon.com if you're interested in participating in what I'm considering a roundtable series. So I have a few ideas of subjects we might want to talk about as a community. But as I said, I'm the life sciences lead. Just so you know, we have leads in the geospatial, climate, weather, and sustainability, and statistical, regulatory, and human systems space as well. So if you don't happen to be a life scientist, but you still want to talk about open data, you probably won't talk to me, but you're going to talk to someone on my team. So thank you very much. Yeah. And I have to talk about digital upskilling, so check out our digital upskilling. And we'll be here for a few minutes to answer any questions.